Good afternoon, everyone. My name is Christianah T, Omotoso, a Master of Pharmacy student at the University of Western Cape under the supervision of Dr. Samuel Guille and Professor Alan Christopher. This afternoon, I will be presenting on the generation of mutated cytochrome P450 enzyme sequences with bioinformatic techniques. Cytochrome P450 enzymes are responsible for the extensive catalysis and biotransformation of most drugs in other lipophilic xenobiotics, and they have a major clinical pharmacological relevance. As shown in the pie chart here, it indicates the number of drugs that have been metabolized by the different isoforms of cytochrome P450 enzymes. Cytochrome P450 enzymes are highly polymorphic and they are involved mainly in phase 1 metabolism of clinical drugs. As seen in the pie chart here, the majority of the enzymes involved in phase 1 metabolism are cytochrome P450 enzymes, and my reference are indicated below. Polymorphism in the cytochrome P450 have the most impact on the fate of clinical drugs in phase 1 metabolism, since almost 80% of drugs used today are metabolized by these enzymes. There is increased rate of reporting the mutation of cytochrome P450, and this necessitates a study. Single nucleotide polymorphism is one of the most common genetic polymorphisms described in the literature and is well recognized in clinical practice. It occurs in sequences of proteins involved in drug transport, metabolism, and in receptors that ultimately influence both the pharmacodynamic and pharmacokinetic properties of many medications. Biologically functional SNPs lead to altered protein structures or expression and are considered the essence and substrate of human diversity in both health and disease. A problem statement: there is a limitation in the availability and accessibility of the mutated sequences of cytochrome P450 enzymes. And our aim is to generate mutated sequences of cytochrome P450 enzymes using bioinformatic techniques. This is actually a step of my main research in relation to the influence of mutation on cytochrome P450 to individualized drug response, that is pharmacogenomics. So the bioinformatic technique used is Python. Python is widely accepted in scientific community because it has very consistent syntax. It's very sensible. It has very sensible set of built-in libraries such as Plumbee, Pandas, and SkyBi. It's easy to learn. It has features such as strings of character that can be well manipulated. It has very clear syntax, and it's also integrated with other existing tools such as Java. Okay, so we're making use of a package in Python called BioPython. It's a freely available Python tool for computational molecular biology that makes Python usage easy with its high quality and reusable modules, and it's also accessible to online services. Functionality is a tool for performing common operation on sequences such as translation, transcription, and weight calculation. It calls for dealing with alignment, including a standard way to create and deal with substitution and matrices, and GUI-based programs to do basic sequence manipulation and translations. My reference here. The method used for manipulation or mutation of a sequences, I downloaded the wild-type sequence from SASBi, which is a database reference, and the sequence downloaded was converted to a mutable sequence because a sequence came as a string that cannot be mutated. So it's the mutable sequence is now edited, and then the mutable sequence is converted after the addition or mutation has been effected to an immutable sequence and is saved back to the disk. I brought out some pictures on my Jupyter Lab notes just to explain further and show the results of my mutation. So there was installation of it by Python. Basically, here, the one result indicates that the mutation had been effected, and as I explained earlier, the mutable sequence was converted to a mutable sequence. The position of the mutation was indicated as I want this particular position to be changed to proline. The amino acid here, I want it to be changed to proline. I actually used a protein isoform here, a DNA sequence or RNA can also be used. Okay, so to do the validation, we used a very simple step in Python, which is known as indexing. Indexing is used to refer to an interval by its position. So we made use of basically an index operator that is being used is a square bracket. So my mutation is being asserted. So in conclusion, we were able to have, there was easy availability of our mutated sequences, which would further our study on the effects of mutation on DNA and RNA variants, as well as protein isoforms in relation to individualized response of individuals to drugs and prodrugs, that is pharmacogenomics. And then this method is quite easy and quick. It's not hindered by the number of nucleotides or amino acids in the sequence, and it's applicable in the generation of other DNA single point mutated sequences aside Cytochrome P450 enzymes for computational molecular biology. I'm grateful to my supervisor, Dr. Samuel Gay, Professor Helen Christopher, my co-researcher, Dr. Samuel Adiboye, and Mr. Peter Van Henderson. Thank you for listening.
Thank you very much, Christianah. That looks like a really good project. Any questions? Okay, George asks, he says it's so cool to combine Jupyter and BioPython. He asks, what are the source of the variants to be used? He's not sure if he missed it.
Okay, the sources of variants to be used are actually gotten from reported articles on variants reported in certain location. So we work basically on that to further study on, okay, as I've said, we're working on pharmacogenomics that is in relation to the mutation that have been reported. So from articles gotten, from reported articles of variants in certain populations.
Okay. George says he thinks you'd find the farm-var site very useful. We have another question from Penelope. She says, how will you test the variants you've generated?
Yeah, I mentioned it in my presentation. We actually how to validate if the mutation had been affected by using a single step in Python, and that's indexing that indicates the position. Okay, would try to validate if the position of the mutation had been affected to check if the amino acid or the nucleotide, as the case may be, it's located in the position we want. So that is done by indexing in Python.
Okay, thank you so much.